Welcome to Biology My Passion. I am Soumya Harikrishna. Today we are discussing a few important questions from the chapter Applications of Biotechnology. This will be helpful for you for your board examination. The Green Revolution has been achieved due to the use of improved crop varieties and agrochemicals, but it is still not enough to feed the growing population. Suggest a suitable method to agriculture to meet the demand. See, actually, we have three types of cultivational practices. One is agrochemical based agriculture, where we use lot of chemicals, fertilizers, etc., which we implemented during the Green Revolution time. Second is organic farming, where little or no use of chemicals during the cultivation, which is safe to use, but the problem is the yield or the production is comparatively less. It takes time. Third is the genetically modified organisms, so which can solve many of the problems. So here the most appropriate answer could be use of genetically modified plants to solve this problem. What are the advantages of these plants? These plants can have better yield. They have tolerance to abiotic stresses, disease resistance, efficient utilization of nutrients, increased product quality, etc. Some cotton plants grown by farmers are known as BT cotton. What does BT stand for? We know BT is the name of a bacterium called a Bacillus thuringiensis. What is the advantage of this cotton plant? Once we are transferring a gene called a crygene from Bacillus thuringiensis bacteria to the plant, then that plant is called a BT plant. So if it is transferred to a cotton plant, it is a BT cotton. This plant becomes insect resistant, very specific to certain insects. How did scientists achieve this? Scientists introduced the crygene from BT, that means Bacillus thuringiensis, to cotton plants using a vector. Usually, for transferring gene of interest to plants, we use the vector called a Agrobacterium tumefaciens. And the bacillus is not killed even after the application of insecticidal protein, which is toxic. Comment. So the bacteria is not affected by this toxin because the cry protein is produced in the form of a protoxin, means in an inactive form. Only when it reaches the gut of the insect, due to alkaline pH, this crystal will solubilize and becomes active. How does the genes Cry1AC differ from Cry1AB? Cry1AC controls cotton bollworms and Cry1AB controls corn borer. They are very specific in their mode of action. Dr. Harsha developed a vitamin A rich potato through her research on genetics. What do you call such potato plant? So it's a genetically modified plant and it's also called a transgenic plant. Who can approve the validity and safety of introducing potato for public uses? Any genetic engineering research should be approved by Genetic Engineering Approval Committee or GEAC. A 4-year-old girl suffered from ADA deficiency. She was cured by inserting a corrected gene into her body. What is this process called? This process is called a gene therapy. In which cells are the genes introduced? These genes are introduced into the lymphocyte cells taken out of her body. Name the vector used to introduce the gene. Usually, for transferring gene of interest to animal cells, we use retrovirus. Hridya was told by her grandmother about the medicinal properties of turmeric. Later, she read an article stating that hybrid variety of turmeric was produced by another country. This new variety was claimed as an invention by them. Do you think it is an invention? Of course not. If it has to be an invention, it should be a novel creation. But here they did only some minor hybridization experiments to make a new variety, so it's not an invention. How could we have restricted the movement of this material? We India has been using turmeric for long, and if we had patented this plant, this would not have happened. What term is used for such an act? This act is called a biopiracy. It means the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. How is proinsulin different from mature insulin? Proinsulin means the insulin produced in the immature form by the body. It has to become mature insulin for bringing about its action. In proinsulin, there are three polypeptide chains, A chain and B chain, connected together by a C peptide chain. Whereas when it becomes mature, it will lack C peptide. Only A and B peptide chains will be there connected by disulfide linkage. Which MNC? MNC stands for multinational company, produced insulin for the first time and how? In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of Escherichia coli to produce insulin chains. Chains A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Now, do you know why they were forced to make this way? Because if you take the insulin gene as such, we would be able to produce only the immature insulin or the proinsulin, which cannot be processed into mature insulin. And also, the insulin produced by this method is called a humulin. List any four advantages of GM crop. GM crop means genetically modified crops. This was already mentioned. More tolerant to abiotic stresses. Abiotic stresses means the drought, salinity, flood, etc. Reduced dependence on chemical pesticides because plant itself is resistant to pests or insects. Then reduced post harvest loss. Shelf life period we can increase means the delay in ripening of fruit and all. Increased efficiency of mineral usage by plants so that whatever nutrients we are giving to the soil will be utilized completely. Enhance the nutrient value of food. We can enrich the nutrient quality like a Atlas 66 wheat or a golden rice where vitamin A is enriched. List the disadvantages of GMOs. Genetically modified organisms have got certain disadvantages also. One is transgenic crops may endanger wild and native species because naturally hybridization can happen, that is cross pollination, and it may lead to loss of wild varieties where we have the original genes present. Or it may cause health problems like by supplying allergens, or it may damage the natural environment also. But still, researchers are being conducted in this field. How is rosy cow different from normal cow? Rosy is a transgenic cow. She could produce milk enriched with a human protein. Her milk had 2.4 grams of this protein per liter. What was that protein? It was human alpha lactalbumin because the gene for producing this protein was transferred to this cow and the milk became nutritionally more balanced so that a human child consuming Rosie's milk would get the milk or nutrients equal to mother's milk. GEAC is one of the new organizations set up by Indian government. Write its full form. GEAC stands for Genetic Engineering Approval Committee. Now give its two objectives. There are objectives like a decision making regarding validity of GM research. Anybody doing research in India should get their permission to get approval for the validity of their research, and also safety of introducing the genetically modified organisms for public use. A bacterium called Bacillus thuringiensis produces a toxic protein named cryprotein that is lethal to certain insects but not to bacteria. Why this toxin does not kill the bacteria, which I already mentioned, that this protein is produced in the form of inactive protoxin, so it cannot affect the bacteria. What type of changes occur in the gut of insects on consuming this protein? Once the insect ingests this protein, the inactive protoxin will solubilize and become active protein. It's actually a crystal protein. It solubilizes to form active form because of the alkaline pH of the gut of the insect. And this toxin will go and bind to the surface of the midgut epithelium, means the inner lining of the epithelial lining of the gut, and it will cause perforations. Perforations means what? Certain holes. Then it will lead to swelling because water will enter and swelling will happen, and lysis means breakdown of cells. As a result of all this, the gut of the insect will get damaged, leading to the death of that insect. So whichever insect is feeding on this particular chemical, they will die. How man has exploited this protein for his benefit. So, specific Bt toxin genes isolated from the Bacillus thuringiensis bacteria were incorporated into several crop plants, like crygene. It was incorporated into crop plants such as cotton and corn, tobacco, tomato, or brinjal, which become pest resistant against certain insects because these crygenes are highly specific or it can act only on certain targeted insects. Name the host plant and its part that Meloidogyne incognita infects. Explain the role of Agrobacterium in production of double-stranded RNA in the host plant. So Meloidogyne incognita is a worm that is a nematode which attacks the root of tobacco plant. As a result, yield is drastically reduced. The Agrobacterium are used as vectors for carrying nematode-specific genes to be introduced into the host plant. So here the vector used for transferring gene of interest to plants is Agrobacterium tumefaciens, and the host plant here is tobacco plant. These genes, when expressed inside the host plant, produces two strands of RNA, sense strand and antisense strand. Since they are complementary, they will form a double-stranded RNA. Later, once the actual infection by the Meloidogyne occurs, the mRNA will be produced by the Meloidogyne, and the RNA of produced in the host cell is complementary to the mRNA. As a result, it will go and bind to the mRNA and silence that process or silence the expression of that mRNA. So this is called a silencing of mRNA, or this mechanism is called a RNA interference. I have done a detailed video of this RNA interference. Those who are interested can go back and watch that video to have better understanding. Why is agrobacterium mediated genetic transformation described as natural genetic engineering in plants? Agrobacterium tumefaciens, a natural pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA. T stands for tumor because it has a plasmid called a TI plasmid, tumor inducing plasmid, which has a gene called a tumor gene. To transform normal plant cells into tumor and direct gene transfer. It can transform tumor cells to produce chemicals required by pathogens. So, tumor-inducing plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic. We removed that pathogenic effect, but is still able to use the mechanism to deliver gene of our interest into a variety of plants. Since Agrobacterium tumefaciens has the natural ability to donate a part of its DNA to the plant during infection, this property of Agrobacterium is exploited and a gene of interest is ligated to tDNA so that it automatically gets transformed into plant cell. Thus, Agrobacterium tumefaciens is known as natural genetic engineer of plants. So these are important questions in this chapter. This chapter is actually easy to learn because there are certain focus area or 3-mark questions only. So what you have to do is either you by heart those or still you want better understanding, you can visit the channel Biology My Passion. Either you can directly search for the particular video or go to that playlist, because chapter wise playlists have been created in both Malayalam as well as English, so you can choose whichever is suitable for you and then watch it. Hope you understood the important questions discussed here. Thank you for watching my video. If you understood the lesson well, please like, share and subscribe to my channel Biology My Passion.